The overall goal of this procedure is to identify potential key regulators of mouse EML cell self-renewal and differentiation by using RNA sequencing technology. This is accomplished by first separating lineage-negative CD34 positive and lineage-negative CD34 negative EML cells based on surface markers. The second step is to isolate mRNA, convert the mRNA into cDNA, and prepare the library for sequencing. Next, the DNA library is subjected to high-throughput sequencing. The final step is to analyze the sequencing results and look for differentially expressed transcription factors. Ultimately, RNA sequencing technology is used to show differentially expressed transcription factors in lineage-negative CD34 positive and lineage-negative CD34 negative EML cells.
The main advantage of RNA sequencing over other gene expression profiling methods such as microarray is that it has higher sensitivity, accuracy, and wider dynamic range, and it does not rely on pre-existing gene annotation information.
This method can help to answer the key questions in the stem cell field, such as what are the key transcription factors regulating cell self-renewal and differentiation.
The implication of this technique extends toward the therapy of blood disease, because better understanding of key regulator controlling the hematopoietic precursor cells, self-renewal, and differentiation can lead to the development of effective treatments.
Generally, individuals new to